Hello everyone, welcome to SRIRAM IAS and I welcome you all to the initiative of Prelims through PYQs. Before this session, we had the session in General Science where we have discussed the questions from the biology. Now this is the part 2 where we'll be specifically looking in the questions from the biotechnology. Now before I begin with the questions that has appeared in the biotechnology section, I just wanted to give you a brief about the biotechnology and its relevance in the UPSC CSA Prelims examination. Now, when you see the questions coming out from the biotechnology section, it has become very important and imperative in the examination part that human diseases and biotechnology has been very dominant in the paper. So make sure that the question we are discussing, you will get a lot of insight that what UPSC is asking in the civil services examination, how the questions are framed. Questions are not on the tougher sides and neither it is tricky. So it is very important for you to get your basic clear, and this is a low hanging fruit where you can easily mark the correct answer. So without any further ado, let's get started with the prelims through PYQs on the science, that is the section is biotechnology. So for the first question for the day that we are looking is, this is for 2024, right? What was the question? It was asked that in which of the following are hydrogel used? So the question is asked specifically about the hydrogens. The statement that is given is it is a controlled drug delivery in the patient. This is the first statement. The second is it is a mobile air conditioning system. It is used in this particular system. The third, it is used in the preparation of industrial lubricant. Now, before you answer this question, you need to understand what are hydrogens, right? So what are hydrogel? Hydrogel are basically a polymer, right? This is a long chain polymer, right? So this is a long chain polymer and it is called hydrophilic polymer, right? So it's a hydrophilic polymer. Hydrophilic. Now what is the nature of hydrophilic? It absorbs a lot of water, right? It has a capacity, it can retain water. Now if you look, I'll show you some diagram that will make things more easy for you to answer this question. So this is the hydrogel. It is used in the contact lenses. It is used in the drug delivery system. It is used in the lubricant and it is used in the tissue engineering. And last but not the least, it is used for the wound dressing as well. Now, the question and the statement that is asked, whether it is used for the drug delivery in patient, definitely it is used there. The second, it is used in the mobile air conditioning system. If you look at the hydrogel, these are mostly used in the batteries and in the other format of the circuits that help to maintain the temperature as such. So this is used in the mobile air conditions. And the third is it is used in the industrial lubricant. Yes, it is used in the lubricant. I have shown you the image. You can see it is used in the industry. It is used as a lubricant. So all the three statements that is given regarding the hydrogels are correct. So your correct answer for this will be D, right? Now moving ahead with the other questions. Now the second question, this was again asked in 2024. You can see this number of questions has been asked and these are very easy questions. The first statement, there are two statements given. It asks that India does not import apples from the United States of America. See, these questions are not explicitly on the domain of biotechnology, but rather it was asked in the context. The second statement is, in India, the law prohibits the importance of genetically modified foods without the approval of competent authority. Now, first looking at the first statement, that India does not import apple. It is not something like that. We have ample productions of the apple. India do import it. I'll show you some data that will help you to understand what are the imports that we are making for the apples. And the second statement talks about, we'll discuss the second one first. Let's take the first statement, right? So the first statement, for the first, you see this is the data for 2016. This was the data for 2016. That we imported from United States of America, we imported from China, Chile, New Zealand, Italy and other countries, right? Now this is for 2022 and 23, right? 2022 and 23, we import from Turkey, we import from Iran, we import from Afghanistan, we import from Italy and Poland. So these are the top 5 countries from where India is making import for the apple. Now coming to the second statement, you must be coming across with the news that we have a competent body when it comes to the genetic modification crop or the regulation of GM crop. That body is known as, this is something that is Genetic Engineering Appraisal Committee, GEAC. This functions under the Ministry of Environment, Forest and Climate Change, MOEF. Now be rest assured, there can be variety of questions coming to you regarding the regulation of genetically modified foods or crop, right? So this is something very important for you to understand on this part, right? So I hope I'm audible, clear for you all people. Now moving ahead, so this is something important body that take care for all the approval, right? So in this, what is the first question? That India does not import. Definitely India's import. So the first and second statement is definitely not correct. Talking about the second, both statements are correct, not. First is correct, second is incorrect, no. The D is the correct answer, that first is incorrect and second is correct. Why? Because we have a competent authority known as Genetic Engineering Appraisal Committee, which is under the functioning of Ministry of Environment, Forest and Climate Change, right? Now moving ahead to the other question that we'll be looking at, we'll be looking at the other question. The third question, yeah, so this was a question which was asked in 2023. So be noted about the time, how the pattern has been changing for the civil services, specifically when it comes to the questions of biotechnology. So aerial metagenomics best referred to which of the following situation? Now what is metagenomics? Before you understand the term metagenomics, you will be easily able to answer what is the aerial in this context. Now metagenomics means the diversity of the organism, the study of the diversity of the organism in the particular part with its DNA, RNA and other specific components, right? Now aerial in this particular means that the collections of DNA samples from air in habitat in one go. For any metagenomics, for any organism, we basically takes into consideration on the study on the part of its DNA and its RNA, right? So for this, the correct option is collecting the DNA samples from the air in one go. This was one of the easiest question that was asked in the civil services examination. The other option is negated, but I'll read it for you. Understanding the genetic makeup of an avian species. This was something to make a gimmick so that you get on a trap. Using an airborne devices to collect the blood sample and the moving animal. Nothing to do with the aerial metagenomics. And the third is sending drones in an accessible area to collect the plant animal samples from land surface and the water bodies. So this is absolutely not the correct one. Now moving ahead with the other question is microsatellite DNA is used in case of which of the following? Now microsatellite DNA, now if you look at the part of the term, this is there, why it is used. See, there are subsets of DNA that is, right? If you look at the DNA component, basically if you look at the chromosome and the part of the DNA, we have A, T, G and C. Adenine, thymine, guanine and part, right? Cytosine. So these are the things which is there. For this, the correct option, let me be the option first, that studying the evolution and relationship among the various species of fauna. The second is stimulating the stem cells to transfer into the driver's functions of the tissue. Absolutely not. Promoting the colon horticulture from the plant propagations and horticulture in the plant. This has nothing to do. Microsatellite has nothing to do with the horticulture part. Assessing the efficiency of drugs by conducting the series of drugs trial and population. This is also not the correct one. The correct option in this is studying the evolutionary relationship among the various species of the fauna. This is why the microsatellite DNA and the part of the technology is being used. Now, moving ahead with the other question, this was asked in 2022. This is regarding the DNA barcoding. Now, this is again something very commonsensical question. You must be visiting to the hypermarts or for shopping. You must have identified that the barcode is specifically used to scan the tag and even to come up with the prices. Now, the same goes for the DNA barcoding. This tool is used for which of the following? There are three statements which is given. First, assess the age of the plant or animal. See, barcoding has nothing to do when it comes to the part of identifying the age. So we'll keep this into consideration, but this is something which is not there, right? I'm considering you to be on the layman's side, so let's consider this. Now, the second is distinguishing among the species that look alike. Definitely, DNA makes a difference, right? DNA and RNA and other components has to be there when it comes to part of the identification, looking alike and making a difference. Now, the third is identifying the undesirable animal or plant material in the process of food. Now, before I explain the answer, let's have a look at the diagram that will make things more clear for you. Now, this is what DNA barcoding is. I think the images would be evident for you to make it understand what exactly it means. So, this is the specimen collection, suppose an example, then the extraction of DNA takes place, right? In that extraction, we have a process called PCR amplification. What is this PCR? PCR stands for polymerase chain reaction. Make sure आप polymerase chain reaction के बारे में भी थोड़ा सा पढ़ ले. This become important for the examination. Now, what is amplification? They actually extract it, right? And after extraction, a sequencing of DNA barcoding is done. Now, this barcoding look like in something and it being repository as a library, and this is how it function. So, for this, the correct option is that DNA barcoding. Definitely, this is not something where we'll actually evaluate the part of age. So, this is absolutely not correct. And these two statements are correct. The correct option here become D. That is the option 2 and 3. Now, moving ahead with the other question. This was again asked in 2022. In context of the vaccine manufactured to prevent the COVID-19 pandemic, consider the following statement. Now, this question seems that why we are making a discussion of this question is something very technical attached to this particular question. So, UPSC might ask you related to vaccines or something like that. So, the first question that is being asked, that the SII, better known as the Serum Institute of India, has produced the COVID-19 vaccines named after Covishield using a mRNA platform. Now, the two things become important, mRNA and DNA vaccines here, right? Now, what is mRNA? mRNA is a messenger RNA platform. Now, this question was asked because COVID-19 was prevalent for a year almost, right? So, it became evidently important for UPSC to ask to understand some basic facts regarding the vaccine. But definitely this option is not correct. Those you have read. This is a platform vaccine, vector vaccine that was used by the Serum Institute of India. AstraZeneca, with the collaborations of Oxford University, has come up with the first mRNA vaccine, not the Serum Institute of India. So, the first option is correct. Sputnik 5 vaccines manufactured using the vector-borne based platform. Definitely, this is the correct one. And the third, we have Covaxin is an inactivated pathogen-based vaccine. Now, what is inactivated pathogen vaccine? See, when the vaccine has been manufactured, there's some inactivated and a low format of the virus is being inserted to express the immune response. जो immune response होती है उसको बढ़ाने के लिए इस तरह के viral response जो हमारे body में immunity होती है उसको बढ़ाने के लिए, उसके responses को बढ़ाने के लिए, these are the concept that is being used, right? So for this particular question, first is absolutely not the correct one. If you go by the elimination, which is not working as of now, तो यहाँ पे जो correct option होगी, these are the again corrected. The two option that is there is basically no. Just allow me a minute. Yeah. So the correct option become 2 and 3, right? So make sure you know the difference between the mRNA platform. This is a messenger RNA platform which is being used, which was used by AstraZeneca. And the other is vector-borne. In vector, what happens that the things like the viruses are actually inactivated form are taken into the human body and it activates the immune response of the human body. Now, something which is there, which is called T cells and B cells. You must be aware of the basics. T cells and the B cells, and in T cells we have T4 lending cell. There are various cells which is there. I'm not diving deep into this concept, but you need to know this particular basics. Now, something other than important is this question was asked in 2022. So you can see the questions coming is large. Biofilm firms, this is something, a statement, you have to consider it. Biofilm firms can form a medical implants within the human tissue. Is this correct? First, you need to understand what is biofilms? See, biofilms has nothing to do with the films, right? It is nothing a part of things that will be taken as a photography. This film means, the biofilm here means that it has a colony of bacteria or the viruses or the microorganism that actually spread in the surfaces, right? I'll show you a diagram which will make things more clear for you. Now, biofilms can form on food and food processing surfaces. The third is biofilm can exhibit an antibiotic resistance. Now, let me show you a diagram that will make things more clear for you. Now, this is how the biofilm actually works. You can see the bacteria or the microorganism attaching to the surface, right? Now, it developed further and it make a certain kind of a colonies. Further, it multiply, and this is what the biofilm is called, right? It has a mature thing which is coming out, which is known as biofilm, or a community of cells coming out of it, right? So now after this diagram, you will be easily answer this particular question, that definitely all three options that is given in the statement are correct. So the correct option here is D. Now moving ahead with the other question is Bolgard 1 and Bolgard 2 technology mentioned in the news is related to which of the following contexts? This is again one of the easiest question that was asked in 2021. The first statement is a colonial propagation for the crop plants. Now this is the first statement. The second is developing a genetically modified crop plant. The third is productions of plant growth and substances and productions of biofertilizers. Now, Bolgard 1 and Bolgard 2 was related to developing the genetically modified crop plant, right? And this was something related to BT cotton. BT cotton was actually there in India. It was introduced in India and way back in 2002, right? And this was by Monsanto, was a company who has actually introduced the BT cotton. Right, and we have also, the government of India has also approved the BT mustard, right? So it is DMH11, right? This was developed by the professors from the University of Delhi. The government has approved with certain restrictions for the genetically modified mustard as well. So there might be the questions coming out from the GM mustards. Now moving ahead with the other questions. This question was again asked in 2021. The question is with reference to the recent development regarding the recombinant vector vaccine. See, the two questions coming from the vaccines, and it might be the case that UPSC might ask you some questions regarding vaccines and other development part. A lot of technological development has been coming up in the human diseases sectors and the advancement sectors. So with reference to the recent development regarding the recombinant vector, consider the following statement. Now what is the statement? The genetic engineering is an applications in the development of this vaccine. Absolutely correct. The second is bacteria and viruses are used as vector. Definitely, not only bacteria and virus, there are various microorganisms that can act as a vector and that can be the part of the recombinant vector vaccine. So in this particular case, both the option, that is option 1 and option 2 are correct. If you want to read more, you can just go on of the part of recombinant vector vaccine, what is all. I have already talked about mRNA, right? So mRNA, that is a messenger RNA vaccine. There's a vector borne. There is something called live attenuated virus vaccine, which is again in the low capacity of virus. The same virus at the low capacity is being introduced at the human body. So do have a check. There might be some questions coming out with the vaccines part. Now moving ahead, with reference to the carbon nanotube, consider the following statement. This question was asked in 2020. Now this type of question, something requires your understanding. The first question is they can be used as a carrier of drug and antigen in the human body. Now for this I have an image which will give more clarity on this part. First let's read the statement first. The second is they can be made into the artificial blood capillaries of an injured part of a human body. See, when it comes to carbon, specifically carbon as an allotrophy, cannot be used as a blood capillary. Now this is something very commonsensical. If you have read, you can easily make out. For this time, we're not considering this particular statement. The third is they can be used in biochemical sensor. Let's consider this statement as well. The third is carbon nanotubes are biodegradable. Yes, this is biodegradable. Now, before coming to the answers, I would like to tell you other CNT biomedical applications which is there. This can be used as cancer cell therapy. This can be used as a biosensors. This can be used as tissue engineering. This can be used as drug delivery, and this can also be used as bioimaging. Now, if questions coming from the carbon nanotubes, specifically in the biomedical application, it has an application in various other domain as well. But if it is categorically asked in the biomedical sectors, you need to understand that these are the things actually working. Now, going back to the statement, now it is asking, I have told you that artificial blood capillaries is something which will not work. It can even have a toxicity effects to the human body. So this answer is absolutely not the one. Now this talks about they have been carrier for the drugs and antigen. Yeah, this is correct. The third is they can be used as a biochemical sensor. Absolutely correct. And carbon nanotubes are biodegradable in nature. So this we have a question. The answer is B, that is 2, 3 and 4, right? So make sure if you able to have a screenshot for the revision purposes, you can have it so that you can revise it well. So this was all the questions that we were about have actually been discussing for the biotechnology part. I'll be coming up with more questions on the human diseases. Most likely that will be the last part of our discussion when it comes to the general science catering the civil services examination requirement, and I have been emphasizing on the part that these are again the low hanging fruit. This require very less effort, giving a contribution of none less than 5 to 6 to 7 question every year. So make sure that you take advantages of all this section of science and technology, which includes biology, basically from the animal diversity part, and the part of human diseases and biotechnology, making a substantial contribution in the biology section. So do have a check on this. Do be regulars once you're reading. Emphasizing on other section will also be important, but you need to understand this fact that these are the low hanging fruit. You need to emphasize on that. On that note, I'll conclude this session. Thank you so much for joining us live.